Okay, well, welcome everyone. I'm assuming you're online and streaming and seeing these slides and myself. It's my great pleasure to actually introduce our seminar speaker this morning, Eric Madsen from the FedHutch, who's doing his promotion seminar to full member. So I'm going to say a few words, but before I get started with a quick intro of Eric, I wanted to let you know that you're going to have the opportunity to ask questions via the chat online, so you can post your questions, and what's going to happen is that they will basically stay there until the end, and then Eric can go through the questions and answer these questions. So if you feel like you have something urgent and you want to ask something, just post the question while you think about it, and then we'll get Eric will get to it at the end. So I'm just going to say a few words, but I mean, I've been a very big fan of Eric. He's been a major contributor of quantitative science at the Hutch, you know, computational biology and data science. He actually trained in mathematics. Initially, he's got his bachelor from Stanford University. Then he actually went on to spend a year in France, where he actually got a diplôme d'études approfondies en mathématiques at the University of Paris 13th. And then he got his PhD in math from Harvard. After that, he did a short postdoc at the University of Canterbury for 1 year, and then did a Miller postdoc fellowship at Berkeley. He's been doing a lot of phenomenal work. He's got numerous awards, including an HHMI Simons Faculty Award. He's extremely well funded. And what I like about the work that he's been doing is that he's really taking a rigorous and quantitative approach to the research that he's been doing. So he's got methods grant from the NIH and the NSF that reflects that. But he's also co-PI on many applied grants, collaborating with people here locally at the Hutch, but also outside of the Hutch. I would say he has sort of two main areas of research that overlap a bit: phylogenetics and studying innate and adaptive immunity through TCR and BCR sequencing. As I said, you know, in either case, he's really been thinking a lot about, you know, rigorous quantitative approaches to trying to solve problems arising in these two fields. And I think, you know, I've been involved in reviewing, you know, sort of review letters for Eric, and I think all the letters that we got were extremely positive. And I think they highlighted clearly that he's been, you know, in many instances, thought-provoking about the work that he's been doing. So an example, a paper that I very much enjoy reading is basically using deep learning approaches to estimate probabilities of TCR occurrences, which is something kind of new, and people hadn't really thought about doing it this way. So sort of a non-parametric approach to estimating these probabilities, which has been very interesting. The other thing is that he's a major contributor to sort of the area of computational biology. For example, he's been writing sort of phyloseminar.org. He maintains a large number of open source software tools that are very well utilized both for phylogenetic and TCR BCR analysis. And also at the Hutch, he's been a major contributor to data science and training. He's been on the internal advisory board for the TDS-RSC. He's been a founder of FedHutch.io. And he's managed to hire a phenomenal leader of FedHutch.io, Kat Herdwick, who's really been thinking a lot about how do we train researchers at the Hutch. And in a time like today, where, you know, some of you might have more time to do training because maybe your lab's been shut down, I think there's going to be phenomenal resources that we'll be able to leverage through FedHutch.io. So all in all, phenomenal work, phenomenal scientists, and I'm looking forward to his talk. So Eric, I pass the baton to you and you can get started. Thank you.
Thank you. Yeah, thank you, Raphael, for that introduction and also for pushing forward my promotion. I appreciate it. Okay, let's get started. So in the talk today, I'll talk about our work concerning pathogens and the adaptive immune system. So the adaptive immune system, one of its main purposes is to recognize antigen. Antigen is a general word for some sort of pathogen-associated molecule. So one of the cell types in adaptive immunology is called B cells. B cells recognize antigen through their B cell receptor. T cells, which is another type of cell, recognize antigen through their receptor called a T cell receptor. And it recognizes antigen after it has been cleaved and presented by MHC. You might be more familiar with the word antibody, which is the secreted form of a B cell receptor. I will use these two terms pretty much interchangeably. So it's an exciting time to study these things because of high throughput sequencing. So we can sequence pathogen DNA and RNA. We can sequence the region that codes for the BCR in high throughput and also TCRs. This can be done on big high throughput machines and little machines that can just plug into your computer. So with this, the question really of our group is, how can we best learn biology using sequence information from pathogens in the adaptive immune system? From a statistical perspective, one of the ways that we like to think about these data is in terms of model-based statistical inference. So just to take a little bit of a metaphor, let's imagine that we have a cannon that has some angle, and then we have some observations. And we're interested in the relationship between the cannon angle and these observations of how far the cannonball goes. So we have some sort of physics model that describes this. So we have some parameters x, and we have a model y. And we can simulate from this model. We can draw data y given some parameters x. Perhaps more interestingly, we can take some observed data y, and we can infer the underlying parameters x. And these two things, simulation and inference, are actually quite closely connected in the sense that we like to choose parameters x such that when we simulate, they generate data that's as close as possible to our observed data. And this is called maximum likelihood inference, if we take the right divergence between our simulated data and the observed data. We like to take this a step further often and think about Bayesian inference, in which one infers not just the best value of the parameters, but all of the basically credible values of the parameters and their probabilities of being correct. So this was, of course, just a metaphor. For our group, typically y is some sequence data, and then x is something to do with pathogens or the adaptive immune system. And so x, I've described a parameter as just being a single real value, such as an angle here, but it can be quite complex. So for us, oftentimes x is something like a phylogenetic tree or something to do with recombination. So now I can phrase what I'll be talking about today. I'll be talking about new methods for Bayesian phylogenetics, which are useful for tracking viruses, new methods for understanding the antibody response, which are hopefully useful for designing vaccines. And then finally, an overview of our landscape of work and some future directions. So I'm just going to pause here and make sure everything looks okay. Okay, seems like things are all right. Okay. Great. So first, new methods for Bayesian phylogenetics. Okay, so this is a fairly familiar diagram right now. People have viruses and they pass them from one to another. And in the process of passage, the viruses accumulate mutations. We can now observe some set of viruses from some set of individuals, and then we can reconstruct a phylogenetic tree that describes, in part, the transmission process that has spread the virus from individual to individual. So I hope people have been paying attention to Trevor Bedford's work and the rest of his group on the nextstrain.org website. So I will go back actually a little bit farther in time and talk about an older paper from 2017, which had to do with Ebola virus. So here, these individuals sampled genomes of Ebola virus through time. That's what these circles mean. And then they reconstructed a phylogenetic tree of the evolution of these things. And this allowed them to make some really interesting inferences about the spread of Ebola virus. And they made this really neat animation that I'll show now. And so in this animation, this is the relevant regions of West Africa. And these are various geographic districts. the district gets edited when there are infections there. And these little comments are showing basically transmission from district to district. So this is a really beautiful and engaging way of showing what's going on with the epidemic. But I want to make the point that basically all of this information is already encoded over here. So this is just sort of going along like a player piano showing what's happening in the phylogenetic tree. So this is a beautiful representation and some really neat work, but I hope somebody in the audience is saying, well, wait a minute, that was just one possible reconstruction of how things could have gone. And that's actually exactly my point. Tree inference is uncertain. So if we think about the range of possible trees on this axis, and this axis being their fit to the data, more often than not, we have multiple trees that have very similar fit to the data. And if we just focus on one tree, maybe we're deceiving ourselves. So the Bayesian approach is to find all of the credible trees and their probabilities of being correct. And this allows us to integrate tree uncertainty into our analysis. So these authors were well aware of this, and they did a beautiful analysis finding things such as the number of viral introductions per geographic location, where they integrate out all the uncertainty in the relevant variables. Everything I've talked about so far is in this paper here. I was not at all part of this paper, but it allows me to make a point, which is the phylogenetic computation behind this paper took 1 month. And that's really not fast enough for being able to control the outbreak based on the insights that we get from this analysis. So the goal of the first part of my talk will be Bayesian phylogenetics at the speed of sequencing. We haven't gotten there yet, but we've made a lot of good progress, I think. So we've gotten pretty obsessed with this problem. Here are the papers that we've published since 2018. And I think we've learned interesting things from all of them. But I'm really just going to be focusing on an approach that's from these 2 papers here. So let me remind you again of the inferential target. So here, this is our posterior distribution that we want to infer. And what that means is we're given a collection of sequences, and we want to infer a phylogenetic tree on all of those sequences. So this posterior distribution means that we want to find all of the credible trees and their probabilities of being correct. People do this a lot. Here are 2 popular phylogenetic software packages. And so it's worth reviewing briefly how these works and the challenges that we have in scaling these up. These packages are based on the Metropolis-Hastings algorithm that is laid down by these 2 papers. So in phylogenetics, that looks like this: random walk Markov chain Monte Carlo. So in this process, one is at a given tree, one randomly selects a subtree, cuts that random subtree, finds a random place for it to land, so a random perturbation, and then accepts or rejects that hypothesis, that proposal, based on what's called the Metropolis-Hastings ratio. And this is set up carefully such that if we let this process run for a long period of time, the frequency with which we visit an individual phylogenetic tree is in proportion to its posterior probability. So this works beautifully, but it has difficulty scaling. And really the reason why this has difficulty scaling is because if we think about the space of all the possible phylogenetic trees, that's a very, very, very small little subset of the set of all possible trees. And therefore, with high probability, we're going to jump off of that in our proposal, and that will lead to a rejection. So this has trouble scaling when we have a large model space. Let's see. I'm seeing some, maybe I should turn off my video. I'm seeing some comments. Sorry. Maybe I'll turn off my video and just. I turned myself off and I turned the video off. I'll just turn off my video. Thanks for the feedback. Let's see. Stop camera. Okay. So I think people can still hear me, but you can't see me. Okay, better now. Cool, thanks. So here we see a problem where this is the number of tips of the phylogenetic tree, And, oh wait, let's see. Thanks for your patience. I can do this now. Okay, great. Where, I'm going to turn this off. Okay. This is the number of tips of the phylogenetic tree, and this is this number of trees on that number of tips. So how many sequences here and how many trees there are. And so with just 50 tips, we have 10 to the 76 possible trees. And this grows super exponentially, which you can see by just the fact that the number of digits is like the log of this size, and it's faster than linear. So this is growing a lot faster than we want. So in my mind, I think that we really need a new approach. And it's 2020. So I can say that in 2020, most people think you have a computational problem, well, you should use a neural network. And people have tried this in phylogenetics, and it hasn't been entirely successful. But luckily, neural networks sit in a larger class of algorithms called differentiable programming. So with differentiable programming, we fit complex models using derivatives. And another thing that sits inside this rubric is variational inference. So in variational inference, we fit rich probabilistic models. So in contrast, neural networks we can think of as being rich predictive models. So this works as follows. Imagine we have a distribution of interest, which is unknown, and we posit some type of variational distribution. In the variational distribution, we have two parameters. It's described by two parameters, maybe the mean and the variance, if the variational distribution is, for example, a normal distribution, which would be sensible in this one-dimensional case. So we fit our variational distribution to our distribution of interest, and then we can use our variational distribution when we're interested in something about the distribution of interest. So if this is a distribution of phylogenetic trees, we can now think of the variational distribution as being a nice approximation for our phylogenetic distribution. So this sort of leads to a question. Let's see, you told me that we don't know this distribution of interest. And so how are we going to fit a variational distribution to it? And there's this just sort of amazing thing, which is that we can actually do this. We can fit the variational distribution without knowing the whole shape of the distribution of interest. And I will talk about that a little bit later. And we will fit these things using gradient descent. So the objective of our variational inference algorithm is to fit our variational distribution to this distribution of interest. And with that, we have some sort of notion of error or loss. We want to minimize that error or loss. And we do so by taking a gradient and using gradient descent. So gradient descent is where we follow the shortest path, well, the most direct path according to the gradient, to this minimum. And this is really important for all sorts of things in machine learning these days, and it allows us to fit, not us personally, but the community to fit neural networks on 1 billion parameters, which is quite amazing. But this raises an interesting question. We want to do differentiable programming with trees, and trees are discrete. How can we possibly take a partial derivative with respect to a phylogenetic tree? And this is really with the problem that I posed to Xing Zhang when he was in my group. Now he is on faculty in China. And he built on previous work from Hona and Drummond and Brett Largett. He did some really beautiful work setting up really the mathematically correct structure for this. And I'm not going to go into that, but I'll just summarize his work by a diagram that looks like this. So this is supposed to be a conditional probability distribution where we assume that we see a certain substructure in a tree here. And we're interested in what is the probability that that structure resolves into a structure that looks like this. So what this is supposed to represent is that we have a phylogenetic tree and there's certain subset of tips here, U. So imagine this is the mammals, and over here, V. Imagine this is the reptiles. And we imagine that we're making our way down the tree and we see, hey, there's a split here between the mammals and reptiles. What this probability distribution tells us is what is the probability that the next split is going to be between these two subsets, U1 and U2, that split apart U. So you could be the monotremes, it's a group that includes the platypus, and then something else, everything else. And so it could be this split or it could be something else. And that uncertainty is encoded in this probability distribution. So now let's just take a little example of this. So now if I tell you all of these conditional distributions, I can tell you the probability of some other phylogenetic tree here, just by breaking it apart. So the first split in this tree is in between A and B and everything else. And then I condition on A and B and everything else. And I evaluate what is the probability that C is the next thing to split off. Then I condition on seeing C. And then I say, what is the probability that D is the next thing to split off? And so on and so forth. So this is a nice way of expressing probability distributions on tree structures. But it's actually not as flexible as it could be, in the sense that we can express an arbitrary probability distribution on tree structures here, but that requires a super exponential number of parameters. This is an approximation that requires merely an exponential number of parameters. So how good is this approximation? So I'm going to show a fairly technical result with a cartoon. So in this cartoon, imagine that we have a big Euclidean space that is parameterized by all of the possible phylogenetic trees. And our target phylogenetic posterior is just a point in this space. Because remember, a posterior distribution is just an assignment of a probability to each one of the tree structures. We can also imagine all of these approximate distributions as being a subspace of this larger space. So here, this is our target, and this is our closest variational approximation. We can also take a Markov chain Monte Carlo approach and approximate this target phylogenetic posterior with another point in the space. And we actually did this by running Mr. Bayes a conservative way, where it was well converged according to the normal Mr. Bayes diagnostic criteria. And the observation that we made is that when we take the closest variational approximation to this Markov chain Monte Carlo approximation, that actually gets us a lot closer to the target phylogenetic posterior than we were if we just used the raw Markov chain Monte Carlo approximation. So that basically tells us that for real data, these types of approximation are actually a nice inductive bias, and they help us calm down some of the stochastic noise that we get with MCMC. Okay, so that is table 1 of this paper. And I guess I should say that this is sort of subtly true here, but it's actually very much true. We can get very much better approximations to our target phylogenetic posterior by doing this. Okay, I'll just pause here to double check and make sure everything looks okay. Okay, good. Thanks, everyone. Okay, we also need to parameterize branch length. So I haven't really talked about branch length at all, but basically it has to do with the length of these branches, and that represents the amount of evolutionary time that's happened along these branches. I'm not really going to talk about it. It's a technicality. It's important. And we had to work on it, but I'm not going to describe that. So this got a little bit technical, but maybe I can bring people back if you spaced out a little bit. How do we do trees with differentiable programming? Well, we parameterize distributions on those trees, and we can take derivatives with respect to those parameters. And that sets us up to do variational inference. So in variational inference, again, we posit some sort of variational approximation to our posterior. So in this case, this variational approximation is exactly what I just told you. It's this subsplit perspective on posterior distributions. We want to do gradient ascent to fit it to the true target that we don't know. And so to do that, we sample from our current approximation, which I've schematized by these little blobs. We can use the sample to form a gradient on the parameters, and then I can update parameters according to this gradient. So now I can answer the question of how do we fit this without knowing the whole shape of the distribution? And just intuitively, we can see here that this point here has a relatively low probability of being generated under the variational approximation, but it's actually quite good according to the target posterior distribution. So what we want to do is change the values of these parameters to scooch this distribution over to increase the probability of generating these things and decrease the probability of generating these things. And so that means that we don't actually need to know the whole shape of the posterior. All we need to do is understand how good this point is versus this point in a relative sense. So this is everything put together, and this work was published in a machine learning conference. We have promising preliminary results. So on this axis is the number of iterations required, and on this axis is the divergence to ground truth. So low here is good, and we can see that in this case, variational inference is quickly finding its way down to a low error, much more quickly than Markov chain Monte Carlo. So this is really preliminary work. I want to emphasize that, and there's a lot of work that we need to do to come up with a full-fledged implementation. And furthermore, this is with a fairly stock way of doing stochastic gradient descent. There's nothing that keeps us from using other alternative training methods that are optimized for the phylogenetic case, and that's something we're working on quite hard right now. So specifically, we're developing a package called libSBN, which is already open source, even though it's under heavy development. And this is our variational inference package written in C++ to do efficient phylogenetic inference. We're using the Beagle library. So we've given it a Python interface, first of all, because people like Python, but also because Python allows us to connect with these other efficient Python interface differentiable programming frameworks, such as TensorFlow and PyTorch. I think this is the first time that these libraries are really being made available in a powerful way for Bayesian phylogenetics. So I'm quite excited about that. I would like to also acknowledge that we're using gradient code from this paper that is unpublished. I'd like to thank two of the main contributors to this package, Matthew and Song. Matthew's in Australia and has been working on time trees, and Song, I'll be talking about his work a little bit later. There are other things that we're trying to do. One is to revive this idea that was popular in 1999, which is this thing called the disk covering method. So in this idea, we don't want to build an entire phylogenetic tree at once. That's too much work. So we want to build an initial heuristic phylogenetic tree, use that to cut up the set of leaves or sequences into small subsets. We build trees on each of these subsets individually, and then we glue them back together. So this has previously been done in sort of a maximum likelihood type approach, but we'd like to do it in a Bayesian sense, where we get a posterior distribution of each of these subsets, and then we glue those things back together in a probabilistic way. And this is work by Michael Karcher in my group, and he's making great progress. We'd also, of course, like to be able to do this for outbreaks. So we'd like to have updatable phylogenetics. So for this, imagine that we've run a phylogenetics algorithm on 4 sequences, and we have a nice posterior, but then we get one more sequence. We don't want to throw this thing away. Instead, we want to incorporate that new sequence in. And the intuition here is that we shouldn't have to do anything for the entire tree. We should just be able to modify the posterior distribution in the region around that newly inserted sequence. And this is work done by Song. And this has been a goal for a really long time. And we did publish a pair of papers about this. But I think that this variational approach is going to be much more powerful. Another observation is it's not really IMC that's the problem, it's the bad proposal distribution. And so why not use our variational inference method as a proposal distribution and then do it Markov chain Monte Carlo? So this is work that Andy McGee is pursuing in my group, and he's also making great progress. So to sum up this part of the talk, we can take derivatives through evolutionary hypotheses. That allows us to use stochastic gradient descent, a powerful technique used to train neural networks, for which there's incredible software and hardware support. And this last point is really important. There are these beautiful libraries out there that are maintained by legions of professional software developers, and also specialized computing machinery that is making these things work. And so I'm really excited that we can bring phylogenetics into the modern machine learning era and use all these new tools. Okay, so hopefully that didn't get too technical. We're going to be talking about some more sort of applied biology type things now, namely adaptive immunology and the antibody response. So just to step back and think about how amazing it is that your body is able to protect itself against all of these pathogens that keep on getting thrown at us. It does so using random generation and selection of antibodies and T cell receptors. So I'm not going to talk about T cell receptors at all today. I'm really mostly just going to be talking about antibodies. So what do antibodies do? One of the things they do is they just bind the part of the virus that it uses to attach to the cell and block it. They do lots of other things, but this is a fine mental model. And the really neat thing about antibodies is that they undergo affinity maturation in germinal centers to optimize their binding to a target antigen. So these germinal centers optimize binding of antibodies to antigen, which I'll just show as this little circle, through an evolutionary process on B cells that happens inside of you. So it looks like this. This is a figure adapted from a very nice review paper. There's a certain specialized type of cell called a follicular dendritic cell, and its job is to display antigen. So as it sort of has antigen on its outside, and the B cell's goal is to pull antigen off of this FDC. And when it is able to pull things off, it gets T cell help, and that gives it the ability to travel down to another part of the germinal center. So in this part, it's able to mutate and divide. The mutation rate is about 1 million-fold higher than the standard mutation rate that we see in cells. And then it's able to go back up for testing. And this is this really beautiful evolutionary process, a true Darwinian evolutionary process that happens inside of you. So the next time you get sick, hopefully not with coronavirus, and you get swollen lymph nodes, you can think about this thing happening inside of you. So if we were God, we would be able to watch this happen in great detail. We'd be able to see every antibody that was being displayed on the outside of the B cell, and we'd be able to see the structure of cell divisions. Of course, we're not God, and so we can only observe a subset of the cells, and then we have to infer what the structure of this is, what the structure of the cell divisions were, and also what these ancestral sequences are. And this is important for rational vaccine design. There are many people right now that are doing the following thing. They observe an individual who has a very good antibody response against a hard to neutralize virus such as HIV. And they find that individual and they say, okay, let's figure out how this individual was able to make a beautiful antibody like this that has such good properties. They use sequences and they do inferences going back in time. And then the vaccine designer's job is to understand how can we stimulate progression of these B cells down this path to get to this really great antibody that we observed. So we have been working in this area with Julie Overbaugh for years now, and the main people we've talked to are Laura and Mackenzie and her group. So Julie's group made this really neat observation a while ago. So she has this cohort of infants taken many years ago. And these infants are able to generate these HIV super antibodies, which are called broadly neutralizing antibodies, because they're able to neutralize lots of different HIV clades, lots of different types of HIV. And the infants are able to develop these antibodies in just 1 to 2 years of age. And that's pretty remarkable. So how are they able to do this? So our contribution to this is Julie's group gives us a whole bunch of antibody sequences, and we build a phylogenetic tree and infer all sorts of ancestral sequences along this tree. And they're able to ask questions like, do these ancestor antibodies bind HIV? And we want to feel confident in our inferences because they do all sorts of experiments on these ancestral sequences that we predicted. So for instance, they'll grow them up in the lab, generate a bunch of these antibodies, put them on the tip of this long glass rod, dip that long glass rod into antigen, and then they can measure the binding of the antigen to the antibody by looking at diffraction patterns of light being reflected off the tip of the rod. This goes even further. We're in collaboration with Kelly Lee's group at University of Washington. They really make detailed dissections of exactly what's going on in terms of binding. So we really want to feel confident in our inferences. So the common approach in the literature is to infer one best phylogenetic tree and the most likely substitution history. And in fact, you see lots of papers that say things like this: we inferred the unmutated common ancestor of the VRC01 lineage. And it's possible that this inference is correct. However, we can't know that with certainty without using a technique that allows us to understand what are the possible levels of uncertainty that we have in these sorts of inferences. And there's actually sort of two interesting types of uncertainty that we have here. One has to do with the phylogenetic uncertainty that I talked about before, and the other has to do with the VDJ recombination process. So I didn't talk about this very much, but basically the way all of these things get started at the root of the phylogenetic tree is this random recombination process. So this random recombination process, one selects a random V gene, a random D gene, and a random J gene. They get trimmed some random amount and get glued together with random nucleotides. So this is another very interesting source of stochasticity for this process. So what we were able to do is come up with a model that basically jointly models both this recombination process and the phylogenetic tree process, and we're able to do Bayesian inference on these things jointly. This was work led by Amrit Dar in my group, but with Duncan and also with Vladimir Menon. And when we apply this to the VRC01 antibody, we see considerable uncertainty. So this is what our Bayesian estimate tells us. And the height of these letters is basically telling us the certainty that we have that that amino acid was the correct one. So we can see that there are a number of different sites that have a high level of uncertainty, and in particular, uncertainty between very different amino acids. So we would expect these amino acids to change the results of binding experiments. All of this wouldn't be possible without Duncan Ralph's work in my group. He's written this program called PARDIS. Here are some various things it can do. Perhaps people will be most interested right now in, he's working on methods now for predicting antibody affinity from evolutionary data, and that should be up on the archive in about a month. Of course, if we really want to do rational vaccine design, we need a really deep understanding of affinity maturation, and that includes both the mutation and selection processes. And I just feel like I really have to give this as a public service announcement, which is just this amazing fact that you have an enzymatic pathway which is purpose-built to randomly mutate your antibody-coding DNA. And that's just really neat because your body has all sorts of processes that make it so that you can keep your DNA intact without having mutation. But here, there's this whole separate pathway that is there to mutate your DNA. And this is called somatic hypermutation. It's known to be a context sense. Oh, whoops. Yes, so I just wanted to say this is an important thing. So this is just a random paper that I grabbed that came through my inbox recently, but they're making claims about a rare mutation. So of course, if we care about rare mutations, we want to have an accurate estimation of mutation probabilities. So we've taken several approaches to this. I'll just be talking about these second two. So it's known to be a context-sensitive mutation process. So for instance, if we're looking at a nucleotide sequence such as AGCTA, the probability that the central C mutates depends on the identity of these flanking nucleotides. So this one, for instance, has a lower mutation rate than this one here. So one of our contributions to this literature is to come up with ways of estimating these probabilities. So if you turn your head to the left, you can see AGCTA. That's how you sort of read off this x-axis. That's this context. And you can look up here and see what is the height here. And the height is basically the relative mutation rate. This is on more or less a log scale. Whereas we look over here and we see that this has actually a lower mutation rate. So there's very stark differences based on the surrounding nucleotide sequence. So our contributions to this work, and this was work led by Jean Feng and David Shaw, and in collaboration with Vladimir and Noah Simon, Vladimir Menon and Noah Simon, is do survival analysis to handle interacting mutations and also regularization. So we were the first to bring regularization to this problem, which resulted in a very significant reduction in parameters and better out-of-sample generalization. So Jean, yes, she's done all sorts of other things. This is just one sort of the many projects she's done, and now she's going to be joining faculty at UCSF. So we were finishing this project, and then this plot, basically not exactly this plot, but this type of plot came across my desk, and I said, wow, we need to restart everything. And this plot concerns something that some authors have called spatial co-localization. And this has to do with what is the probability of seeing a mutation at I and I plus D compared to the probability of seeing those two mutations independently. So for instance, what this plot is showing, so this is the distance between the sites, and this is the spatial co-localization. So for instance, this point here that's about at 20 and 2 means that if you look at a pair of mutations that are 20 bases apart, those are twice as likely to happen as each of those mutations happening individually. So there's real clustering happening in terms of these mutation processes. So this is a really, this is a very nice oh yes, and to compare that are these little local models that I was talking about before. Of course, this spatial co-localization drops off very, very quickly. So if you just are looking at a 5-mer model, you can't have any correlation past 2 bases on either side. So this is a problem. And I would really like to acknowledge this nice paper by the Mora-Volchak group where they pointed this out. And if you look at the biochemistry, it's not too surprising why this sort of thing would happen. And that's because somatic hypermutation happens in this complex way where initially there's this mutation happening by this enzyme called AID, and then that gets resolved in a number of different ways. And one of the ways in which it gets resolved is this Pac-Man thing comes in and chews out the other strand, and then along comes an error-prone polymerase and introduces mutations in the region of this original lesion. So it's not too surprising that we see clusters of mutations. So we decided, okay, well, if this is the real thing, what we want to do is parameterize a model that looks actually like this, a true biochemical model that comes from this established mechanistic model. This is a hard project. And one of the reasons it's really hard is that there's lots and lots of different ways for this process to give rise to an observed collection of mutations. There's such a large number, we can't calculate something called the likelihood, which is what we would really like to be able to calculate in order to infer parameters for a model like this. And when I say parameter, I mean like, what is the probability that if we see an AID lesion that it gets repaired by one of these 3 pathways? How long does this thing get stripped out? And so on and so forth. So those are the sorts of parameters I would like to have for this model. And so this project is being pushed forward by 2 very courageous people, Julia and Thayer. And they are developing a way to use neural networks to infer, basically, you take a sequence and you infer what is the process by which we think this process happened, but we have to do it in a probabilistic sense. And then using that information, then we can use more classical techniques to infer the parameters of this model. And then we're training it using iterative simulation. So this is nice work. It's a very hard problem, and we're still working on it. And I don't think I mentioned that this is also joint work with Noah Simon. Okay, so just a few more things. The selection landscape is also very important, and we've thought about that in a couple different papers. I'm just going to talk about something I'm really excited about, which is work by Tyler Starr. Tyler is a postdoc with Jesse Bloom and myself. And Tyler is doing a deep mutational scan of VRC01, which is this, a VRC01 class antibody. So in a deep mutational scan, one takes a protein like this and tries substituting every single other alternate amino acid at every site. So Tyler was able to do this for VRC01, and this is basically a summary of the results. So the scale, if we're going from blue to yellow, so yellow means that it's very sensitive to mutation. So this is giving a really interesting and detailed perspective on the fitness landscape for this thing, and that should tell us about natural selection on the lineage leading to VRC01 and other related antibodies. So I'll just give a little bit more overview. Maybe I'll just double check to make sure nothing has gone horribly awry. Okay, great. Cool. So here's an overview of a few more things. Will, in my group, has been working on germinal center dynamics, and plus other means he has this whole other separate research program with Kelly Harris where he's doing amazing things. Eli has been working on antibody lineage visualization. Brandon has been working on immune repertoire comparison. So an immune repertoire is the ensemble of adaptive immune sequences that we have, and somatic hypermutation. So I can say a little bit about what I'm excited about for the next 5 years. I'd like to work with Julie more on HIV antibodies. I'd like to work with Phil on T-cell repertoires. I didn't talk about this work at all, but we've had a lot of fun, and Phil is just a fantastic collaborator. I'd like to work more with Jesse on modeling protein fitness using the sorts of experiments he is so good at doing. Work with Leslie Gu, who's a relatively new arrival on dengue antibodies. And finally, I'd really like to scale Bayesian phylogenetics for epidemic control using the methods that I've talked about with Alexi Drummond, Matthew, Mark Suchard, and Cheng, and other people in their group. So I mean, especially now, I'm very, very motivated to pursue this. And this is going to be a lot of coding that will set our keyboards on fire. And in a larger sense, I'm very interested in this model that I've talked about a little bit here. So differentiable programming is, I think, a nice general way of thinking about the modern machine learning toolkit. And it's really at its core about function compositions and their derivatives. And that's not what biology is about. Biology is about hypothesis-driven research, evaluating hypotheses in the light of data and mechanistic knowledge. And I want to keep on thinking, and I hope the community, we as a community can keep on thinking, how can we drive forward these goals using these new tools? So 5 years ago, this was our logo. We start with biological questions, prove theorems, and make algorithms and write software and hopefully answer the questions. This is still basically true. We do a little bit less combinatoric than we used to, but this is basically the case. But this is sort of a boring logo, so we have a new logo. This is our new logo. It is a child riding a robotic chicken holding a pencil. So the robotic chicken is meant to symbolize computers that aren't very smart, but they run very fast. The pencil is meant to symbolize math, and this is us having a lot of fun charging forward and trying to go interesting places in biology. So that's all been pretty fun. I guess I want to end on a bit of a somber note. In the past few months, I think that we've been acutely aware of the fact that science can warn of and guide action against threats, and also that sometimes radical government intervention is necessary. And this is still absolutely positively true for the coronavirus outbreak. And I hope we can keep on pushing these messages, because we really still need more radical government intervention in the United States. And I don't want to belittle the importance of the coronavirus epidemic, but I do also want to point out that climate change is an even bigger problem, and it is even more slow moving. And we need to keep thinking about these and pushing these messages to the people who will listen. With that, I will stop and say thank you very much to my family, my wife, my little toddler, my parents, and my sisters. Thank you so much for your support. My group, thank you so, so much for all your beautiful work and companionship. The admins, Melissa, Sam, and Ruby, who make it so that I can just focus on my work only. Thank you so much. Scientific Computing for keeping things running and giving a whole lot of scientific computing power for us. Julie has been a fantastic collaborator and so supportive through the years. Phil, Jesse, and the rest of the Comp Bio faculty for being great colleagues and collaborators. Vladimir Menon and Noah Simon, who are really super important collaborators statistically for us in the past and in the future. Matthew and Aaron in Australia for collaborations about phylogenetics. We have basically a new collaboration with Mark Suchard's group, so I'm grateful to them for that. And we're also working with Christian Swampool and Alexi Drummond on a variational inference as well. I'd also like to thank the National Science Foundation and the National Institutes of Health and HMI Simons for supporting me. And also a few more thank you to the healthcare workers who will be treating some of us, no doubt. Researchers who have really put down everything to try to understand and control this disease, especially Trevor, Betts, and Keith at the Fred Hutch. And I just have to tell a little story about Trevor and how grateful I am and the impact that he has had. So there is someone, I don't know who it is, who has my phone number and is texting me. It's a wrong number, and I've told them it's a wrong number, but they keep on texting me. They text all sorts of crazy things. They text things such as, I bought a lot of toilet paper. I think I'm going to make 100K gouging people on toilet paper. And they go on to say, I wonder if Andrew Carnegie or Henry Ford started this way, by gouging people on toilet paper. I think the answer is no. But that's not the point of my story. The point of my story is this, that the next thing they link to is this result. So if we go to this Gizmodo page, it'll probably just have all sorts of pop-ups. But basically, if we look, it's talking about, well, it's re-reporting something that was in the New York Times. And it's quoting Trevor Bedford. And that's just amazing. I will never have the sort of impact that Trevor has had. And I'm so, so grateful to him for doing this early detection and warning work that he's done. And I mean, if your work is being read by the toilet paper gouger, you're having true impact. I'd finally like to thank my in-laws for hosting me for the past few weeks and taking care of my toddler. So I've continued to be able to get things done. So that concludes my talk. If you want to ask a question, I encourage you to do so. So next to this video, there should be something that says chat. And please go ahead and type your question there. So hopefully everything is still going well. Maybe I'll turn my video back on. Okay. So Nina Salama asks, is there any evidence that the type of antigen influences the probability landscapes of the different possible mutation processes in hypermutation? Bacterial versus viral, protein versus carbohydrate. So that's, oh wait, hold on. Maybe I'll just try that. Ooh, that's rather big. How about that? So that is an interesting question, Nina, thank you. But I guess I would say that the mutation processes really, I'll just leave there. The mutation processes should be constant. The mutation processes are really just a function of the underlying biochemistry. What does change a lot is the selection processes. So whatever it is that the antibody is sort of trying to optimize towards, that is going to control the selection. I mean, I guess I can't exclude the possibility that there's some, I mean, you could imagine that maybe some toll-like receptor or something is releasing some cytokines and that changes the distribution of mutations. I guess maybe that's really what you're asking. That's a very interesting idea. I hadn't thought about that, but perhaps. For now, we really have our hands full just trying to understand the somatic hypermutation process as it is without sort of some further modulators. But I think it's sort of an important point to make that when you get a vaccination, you're not sort of stimulating a new VDG recombination. And I don't think that probably the somatic hypermutation process really knows. It's really just paying attention to what it needs to optimize towards. Okay. I think can somebody just post something to make sure that I understand? Okay. Yes, okay. My Uncle Bert, Albert Meisenbach, has asked, why do you care about past phylogenetic history of a virus if you are dealing with a virus as it is now? And that's a phenomenal question. So we can go back. So this is Well, how about this one? There's several different answers to this question. So one is we want to be able to understand things about how the virus gets transmitted. So this, for instance, they were able to infer the number of introductions per location in this paper. And that basically tells them, if we're looking at a map, how much should we work to try to control travel between these various locations. And so that there's just sort of, this is the way that we learn about the evolution, about the history of how the virus gets transmitted around. We can do that using sequenced genomes. And that will help us inform our viral control strategies. And as we know, these are important. There's another answer, and that is that there's also a predictive aspect. So if we can understand the evolution that we've seen so far and the transmission that we've seen so far, we would like to be able to predict where we should be worried about the next, where things spreading more, and also about what lineages of the viruses are more likely to spread further. So Betts says, when do you envision contributing to epidemic control beyond what we already have? So also a great question. So I want to really emphasize that Trevor has done a phenomenal job with Next Strain. And I think, I think that perhaps what we'll be doing is more icing on the cake. But let me just tell a story that, so Trevor did this really nice back of the envelope calculation recently that had to do with, if we see two similar viruses that appear in a similar geographic location, what is the probability that those are two independent introductions, or did the virus just mutate a little bit? And like I say, he did a great back of the envelope calculation, and maybe that's all that we need. Personally, I would like to be able to have something where we could evaluate a probability, where we could say, with probability X, we believe this to be true. And it's sort of stronger than a back of the envelope calculation. So what I'm imagining is that we would be able to come up with a posterior probability that would tell us what is the probability that these two things came from community transmission versus two independent imports. And so in order to do that, we sort of need to boot up the entire Bayesian framework that allows us to integrate out the uncertainty in all the variables. I hope that answers the question. Let's see. Have I seen... Trevor says I would too. Oh, thanks, Trevor. Raphael asks, have I seen the adaptive and Microsoft announcement to decode COVID-19 immune response? They will make their data available. Any thoughts on how you would contribute? Nope. I kind of assume that there's plenty of people working on that right now. But that said, if anybody has an idea about how we could contribute, I would love to help, of course. I mean, anywhere we can contribute, I would love to hear about it. My impression is that, yeah, well, I don't actually know anything about coronavirus immunoresponse. Let's see. So will variational phylogenetic inference become available in BEAST as part of your collaboration with Alexi Drummond? That is the long-term vision. Maybe it's, BEAST 3 but I want to emphasize that this is way down the road. BEAST is this incredibly complex software package with many, many models. And what I've talked about is really ambitious. We're trying to basically take the entire engine out of BEAST and replace it with something rather different. BEAST has been around for many years and it has gotten very, very mature. But that is definitely the long-term goal. But for the time being, we're going to be having Markov chain Monte Carlo. And they have continued to work on how can we scale Markov chain Monte Carlo as it is for epidemic response, such as sort of using parallel. But I think the fact that Alexi and Mark Suchard are interested in this project and are actually working on variational inference with us tells you something, which is that even they don't think that Markov chain Monte Carlo is ever, even with all the fancy things that we can throw at it, is ever really going to scale without some new ideas. Okay, well, let's see. I don't see any other questions, so I can say thanks to everyone for all their questions. That was fun. I hope it all worked out. I'm just going to look up. I don't see anything else. Okay. All right. Thanks, everyone. I'm going to end this now. This video will stay up on YouTube, so all of my ridiculous comments will be immortalized forever. Bye now.